Now we will discuss how the class 2 molecules work. I told you in the beginning of the lecture that class 2 molecules are specially expressed in antigen presenting cells, like they are expressed in the macrophages, they are expressed on the surface of dendritic cells, B cells, Langerhans cells. All these cells are professional antigen processing and presenting cells, right? Let's suppose we talk about a macrophage. Macrophage express class 1 and class 2 molecules both, right? Because they are nucleated cells, they do express class 1. Because they are antigen presenting cells, they also express class 2 molecule. Is that right? So let me make a nucleus here with short arm of chromosome number 6, and let's suppose these are the genes for class 1 and these are the genes for class 2. Is that right? And in class 1 molecule, I have told you already that class 1 molecules are synthesized by the gene A, B or C locus, and class 1 molecules eventually fold them into alpha 1, alpha 2 and alpha 3 component, and they get associated with class 1 molecule with beta 2 microglobin, and class 1 molecule in their pocket antigen binding sites, they bind a fragment of a peptide which is being synthesized within the cell, which is our own endogenous protein. Is that right? And eventually this complex will come on the surface of the cell. So this is the class 1 molecule. After that I will go to class 2 molecule. It is being expressed on the cell, and you already know that this antigen, if its endogenous antigen is recognized by T cytotoxic as non-self protein, then you remember which cell will attack this area? T cytotoxic. We discussed in previous lecture that TCR of the T cytotoxic will bind with the peptide presented by class 1, and it will recognize the class 1 through CD8, and this was TCR. And then you remember the target cell will be attacked by perforins, by granzymes and by Fas-Fas ligand interaction. This was about class 1 action. Now we come to the class 2. And another important thing you must understand that class 1 molecule was assembled right within, what is this? Endoplasmic reticulum, right? The class 1 molecule meet the peptide fragment within endoplasmic reticulum. Now we come to the concept of class 2 molecules. Actually, class 2 molecules bind not with the endogenous protein. How it happened? Let's suppose class 2 gene express, right? When this gene will express, it will synthesize class 2 molecule, right? Let me make a class 2 molecule here. Once again the basic principles are same. Gene will express, it will make messenger RNA. That messenger RNA will be translated into peptide chains, and those peptide chains will be going into endoplasmic reticulum. So actually, if these are the ribosomes and messenger RNA is on that, then class 2 molecule peptide are synthesized and they are also pumped into endoplasmic reticulum. But the very important thing is class 2 molecule consists of 2 peptide chains, and both of the peptide chains are having their genes in MHC locus. I told you class 1 was having one big peptide chain with a small beta 2 microglobin, and I told you in class 1 one big peptide chain, right, alpha chain, was coded into MHC region, but beta 2 microglobin was not coded in the MHC region. But when we come to the class 2 molecules have 2 peptide. Let's suppose this is class 2 molecule, 1 peptide, it does have one more peptide. So it is 2 peptide molecule. And both peptide chains, alpha chain and beta chain, alpha and beta chains of the class 2, both are, of course they are held together, both of them are having their coding genetic system within the MHC locus. You understand the difference between class 1 and class 2? The class, secondly, another very big difference. I will repeat it. Look, class 1 molecule has alpha chain and beta 2 microglobin. Class 2 has alpha and beta, right? In class 1, this whole is called MHC complex on the chromosome number 6. MHC complex is responsible for class 1 proteins as well as class 2 proteins, right? Now, as far as class 1 is concerned, class 1 alpha peptide has its genetic coding in MHC region, but class 1 associated beta 2 microglobin does not have its coding in MHC complex. It is on chromosome number 16, far away from chromosome number 6. Claro? Clear? Now we come to class 2 when we compare this thing. Class 2 molecule consists of alpha and beta chain, and both of them are coded by the genes within MHC complex. So class 2 do not have beta 2 microglobin, they don't have any other protein with them which is coming from some other chromosome instructions. Clear? Now, another thing, that antigen binding pocket of class 2, suppose this is the class 2 molecule. Class 2 molecule has, what is this? Alpha chain and beta chain. And here we have our friend, because we are very familiar with it, which is class 1 molecule. Now, the difference is class 1 has 1 MHC component, class 2 has 2 MHC component. Do you understand it? It's so easy to remember. Class 1 has 1 MHC derived peptide. Class 2 has 2 MHC derived peptide. Is it difficult? 1 has 1 and 2 has 2. It is a little beta 2 microglobin just to support it. Right? Clear? Then another very important point. Antigen binding sites in class 1 is a pocket within one peptide. This is one peptide. All. So antigen binding pocket, if antigen binds here, right? Antigen binding pocket is, or antigen binding groove is a special sequence of amino acid within one peptide chain. Beta 2 microglobin does not participate in formation of the antigen binding sites. Clear? Class 2 should have 2 peptide, both of them participate in antigen binding site. Suppose this is the antigen. You see, antigen is clamped by 2 peptide. Here antigen is clamped by 1 peptide fold. So again, antigen binding site of which MHC molecules has contribution by 2 peptide? Class 2. Is it difficult? I don't know why people find it difficult to remember. Is that right? Another thing, class 1 molecule will bind only with the endogenous proteins, proteins which are normally synthesized in the cell. But antigen presenting cells capture some extracellular proteins. Antigen macrophage may capture some viral protein, may capture or trap some bacterial proteins, may trap some other external proteins which are called exogenous proteins or extracellular proteins. Is that right? Some antigens are present. For example, here the bacteria, and this is bacterial antigen which is exogenous or extracellular antigen. But if virus is synthesizing this protein within the cell, then viral antigens are endogenous protein. Is that right? So antigens which a cell has to present may be endogenously synthesized, or antigens which a cell has to present may be captured from outside. Class 1 molecules are designed to bind with endogenously synthesized proteins fragment, as we discussed in detail previously. Class 2 molecules duty is not to bind with endogenous peptide fragment, because that work is done by class 1. Class 2 molecules duty is, the work of the class 2 is to bind with those antigens which antigen presenting cells are captured from outside the cell, which are exogenous antigens, exogenous peptide. Is that right? For example, this cell captures a bacteria and this is bacterial protein peptide. Of course, this will come over here and lysosomes will bind here, and these lysosomes will digest away break down the bacterial fragment, and this is bacterial antigen which is exogenous antigen, but this antigen is present in the endolysosome. Do you think this antigen, bacterial antigen, is present in endoplasmic reticulum? No. When macrophages or B cell or any other antigen presenting cell like dendritic cell or Langerhans cells, when these cells capture exogenous antigens, they internalize the exogenous peptide into lysosome or endosome. But endogenous peptide fragments are present in endoplasmic reticulum. So it means class 1 molecule which are supposed to bind with endogenous antigens, they will meet the endogenous antigen in the endoplasmic reticulum. But class 2 molecules are supposed to meet with the exogenous antigens, so they will meet them in endosome. It means class 2 molecule need to be transported to the lysosome and endosome. But we have to take care, while class 2 molecules are, let's suppose, transferred to the, what is this, endosome, while class 2 molecules are going towards the endosome so that they may find some exogenous captured antigen, while they are on the way, there should be some special care that some endogenous antigen does not fit in. Have to be their care or not? Yes. Because originally class 1 and class 2 both molecules are synthesized within the endoplasmic reticulum, right? But class 2 molecule, we don't want that these molecules stick to any endogenous antigen. So what we do? On its mouth we put a class 2 molecule passes through endoplasmic reticulum like this. There is a special peptide chain which is covering class 2 molecule on its mouth. This is called invariant chain. What is it called? Invariant chain. So what is invariant chain? Invariant chain is a special peptide which is present on the antigen binding site of the class 2 molecule when it is passing through the endoplasmic reticulum towards the endosome or lysosome, so that some endogenous peptide fragment does not bind here. Once class 2 molecules are transported, where? They are transported into, yes, into endosome, right? Then invariant chain undergo proteolysis in acidic environment. When it undergo proteolysis, now class 2 molecule has opportunity to bind with any fragment which is nearby and fragment which is complementary to this antigen binding site. So class 2 molecules are constantly coming, and again in the class 2 genes also there is one region which is constantly shuffling. In class 2 genes also there is one region which is constantly changing, rearranging. Due to that reason, every class 2 molecule may have slightly different antigen binding site than other class 2 molecule. So if this is bacterial antigen, sooner or later a class 2 molecule will come which will bind with this specific bacterial antigen. Once class 2 molecule bind with this bacterial antigen, then this complex will be brought on the surface. This is one peptide alpha and this is suppose beta peptide, and these are its constant regions, these are its constant, suppose this point is constant region, this area is constant region, and this area is variable region where what will bind? Antigen will bind. Let's suppose this is the antigen which is binding to class 2. Is that right? Now, how it really works, that sooner or later this macrophage, which is expressing exogenous antigen in association with class 2 molecule, sooner or later this macrophage will find such T cell which have such TCR which could identify this antigen. What is this? TCR, which can identify this antigen. Right? Once antigen binds with TCR in association with class 2 molecule, then this T cell will confirm that is the antigen fitting into TCR is appropriately presented with class 2 molecule, and this molecule is called CD4. So what did we learn? And such cells which have CD4 molecules are called T helper cells. So what we have learned, that T helper cell recognize a peptide antigen in association with class 2 molecule, and T cytotoxic recognize antigen in association with class 1 molecules. It means activity of T helper cell is limited or restricted to those peptides which are presented with class 2 molecule. Activity of T helper cell is restricted to only those peptides which are presented in association with class 2 molecule. That is why we say the T helper 2 function is MHC class 2 restriction. So there is MHC class 2 restriction. If some antigen comes with class 1, T helper cell will not respond. So the T helper will not respond. Opposite to that, T cytotoxic activity is restricted to those peptides which are presented in association with class 1. So we say T cytotoxic are MHC class 1 restricted, and T helper cells are MHC class 2 restricted. Are you understanding? Am I clear? For example, you are looking for a girl and you want a girl who should have 5 kilograms of gold. So we say Mr. Lewis girl choices 5 kilogram gold restricted. Is that right? In the same way, T helper cell activities MHC restricted. Which MHC? MHC class 2 restricted, right? And T cytotoxic activities MHC class 1 restricted. How to remember it? T helper is CD4 into class 2 and CD8 into class 1. The multiplication should be equal to 8. It's easy way to remember. The CD4 should interact with, in association with class 2 molecules, and CD8, to keep it 8, should interact with those peptides which are presented in association with class 1 molecule. You have a question?
Yes. The macrophages or antigen presenting cell present this antigen to T helper cells in the spleen and lymph nodes only, and then
No, anywhere.
It can be.
Look, if I get infection here, some bacteria are caught by the macrophages here. Bacterial antigens will be expressed in this area in association with class 2 molecules to T cells, or macrophage will drain into lymph node and then they will find and interact there.
Okay, so both places.
Yeah. Is that clear? Now, having said all of this, let's compare class 1 and class 2 molecules for the last time. Number 1. Class 1 molecules are expressed in every cell, every nucleated cell and platelets. Class 2 molecules are expressed only on the membranes of antigen presenting cells. This is one difference. Second, class 1 molecules bind with endogenously synthesized peptide fragments. Class 2 molecules are designed to bind with the exogenously captured antigens or extracellular peptide fragments. Another difference. Class 1 molecule has antigen binding site in the folding of a single alpha peptide. Class 2 molecules have antigen binding site which is contributed by alpha chain as well as by beta chain. Is that right? Class 1 molecule has alpha chain coded by the MHC complex of genes, but beta 2 component coded by the gene on chromosome number 15. Class 2 has alpha and beta both genes which are genetically coded in MHC complex system. Is that right? Then another thing. Class 1 molecule present antigen to T cytotoxic cells. Class 2 molecules present antigen to T helper cells. Is that right? Do you have any more question? Anything you want to know? Now, having said all of this about class 1 and class 2 molecules, now we will briefly mention what are the biological importance of class 1 and class 2 molecule. We will sum up the whole thing and make some clinical sense out of that. What are the importance of understanding class 1 and class 2 molecules? Class 1 and class 2 molecules, there are three biological importance concepts. Biological importance of MHC molecules. Right? What is the biological importance? There are three important things. Number one, they play a role in antigen presentation. Class 1 and class 2 molecules play a role in antigen presentation. We have discussed into detail. But you have to remember, class 1 and class 2 molecules only present peptide fragments. Is that right? They only express peptide fragments, number one. We have discussed into detail how class 1 molecules present the endogenous antigens, endogenous peptide, and how class 2 molecules present the exogenous peptide antigens. Clear? So this is one biological important. Second biological importance thing is that role of HLA system, or we can call it or MHC system in autoimmune diseases. Let me explain. For example, it is well recognized clinical finding that HLA B27 is strongly associated with an autoimmune disease called ankylosing spondylitis. Ankylosing spondylitis is an autoimmune process in which autoimmune system lead to inflammation of sacroiliac joint and also inflammation along the vertebral column joint. Is that right? Now, or many people know that HLA DR4 is associated with rheumatoid arthritis. How it happens? For example, many doctors know that patients with HLA DR4, right, are more associated, they have more risk to suffer with rheumatoid arthritis. How this particular class 2 molecule, HLA mean MHC, isn't it? HLA D region 4 R4. You may have DR1, some people have DR2, some people have DR4, some have DR5, so and so forth. Out of the whole population, people who are having HLA DR4 gene, they have high risk to suffer with rheumatoid arthritis. Why? Or people who have class 1 molecule of B27 type, they have more chance to suffer with ankylosing spondylitis. Should I explain it to you?
Yes.
What is the relationship? Let me tell you. Initially, it is our observation that in rheumatoid arthritis, autoimmune process attack against the synovial membranes. Is that right? And against the connective tissue. There is some hypothetical theory, that probably in rheumatoid arthritis what happened, we know immune process is attacking the connective tissue antigens. Suppose this is connective tissue, and in this connective tissue there is a special antigen, and against this immune system is attacking. Maybe this antigen is present in synovial membranes. So up to now the basic theory is I will discuss rheumatoid arthritis in big detail in some other lectures, but right now I'm saying that we say the people who are suffering with rheumatoid arthritis, they have immune process directed against some connective tissue antigens. At the same time, now doctor believe there is some microbe in the environment, and that microbe has an antigen, which is very similar with our some connective tissue antigens. Doctors believe that when this microbe come in our body, and if this microbial antigen trigger immune response, then immune response not only goes against the microbe but also against our own tissue, because our own tissue antigen and microbial antigen have molecular mimicry, molecular similarity. Are you understanding? What happens? They think probably some microbe or some antigen comes in our body, foreign antigen come in our body, activate our immune system. In ideal world, immune response should react only against this. But unfortunately, because this antigen is very similar with connective tissue antigen, so immune response not only fight against the foreign antigen, but cross react with our own connective tissue antigens and precipitate rheumatoid arthritis. Are you understanding? Now what is the whole this talk as, what it has something to do with the MHDR4, I will explain. Now they say that this environmental antigen can only fit into those class 2 molecules which are produced by the people who are DR4. For example, there are 3 people. This is DR1. There is another person who is DR4 and another person who is DR5. These are 3 human beings. When we say this person is DR1, it means in the class 2 molecules which are made by this on the D region in the R locus belong to which allele? 1 allele. Here they belong to allele type 4. Here they belong to DR allele type 5. Now they discovered that actually the class 2 molecules which are synthesized by DR4 positive human beings, those class 2 molecule very efficiently bind with this antigen. So it means people who are HLA DR4, they more efficiently bind with this foreign antigen if it ever comes to them. So it means their immune system is more ready to attack against such antigen. So their immune system has more chance to produce rheumatoid arthritis. Opposite to that, if someone is DR1, he does not have a big risk of developing rheumatoid arthritis. Why? Because people who have DR1, they produce such class 2 molecule which do not process this antigen. If a person is not processing this antigen, he is not going to trigger an immune response against this antigen or cross reaction against the connective tissue. So such person does not have a risk of, has a less risk of rheumatoid arthritis. Could you understand that by inheriting a specific type of MHC genes or HLA genes predispose you to a special type of autoimmune disease? Now we repeat the same concept with another thing. We know people who are HLA B27 positive, they have a high risk of ankylosing spondylitis. Now let's suppose you have 2 friends, and this is another friend. He has HLA B27 and this person has HLA B not 27, maybe B6. Now the person who has inherited HLA B27, from the B27 locus, he will make slightly different type of class 1, because ABC is class 1. He will make slightly different type of class 1 than the person who is having HLA B6 inheritance. Is that right? What they discovered, that in the sacroiliac joint and spinal, vertebral joints, they have a special type of antigen. They have very special type of antigen. This special antigen is present in the sacroiliac joints. This is our normal antigen. Of course, this antigen is present in the person who is HLA B27. Also, this antigen is present in the person who is B6. Now we say probably there is some microbe which has similar antigen. But this microbial antigen is more efficiently, just a minute, this microbial antigen is more efficiently taken up and processed with the HLA-B27 molecules. But you have to remember, HLA-B27 molecule normally bind with endogenous antigens. But here, because this bacteria or its antigens go within the cell, and this bacteria or microbe multiply within the cell, so they become endogenous. Am I clear? Now, so is this microbial antigen is more efficiently processed by class 1 molecules belonging to B7 category. But this same bacterial antigen is not properly and efficiently processed by HLA-B6 antigens. You understand it? Now, so this person will develop immune response against this antigen, and that person will not develop immune response against this antigen. But because when this person will develop an immune response against this antigen, because this antigen is very similar with sacroiliac joint connective tissue antigens, it will produce sacroiliitis. The term which is used is sacroiliitis. So such persons who are belonging to HLA-B27 group, they are more vulnerable to develop ankylosing spondylitis. So understanding of HLA system or MHC system should help you to understand why different people with different MHC molecules have predisposition to specific type of autoimmune diseases. Am I clear? Really clear? I'm surprised. Yeah, sometimes people don't understand it. The how HLA system, I think there will be doctors will be really merciful to you if they call HLA system now MHC system. Now the last thing, the third clinical importance, the biological importance of MHC molecules in transplantation. I've already discussed into detail this importance, right? That it is very important that if in the recipient and donor MHC molecules are matching, is that right? Chances of rejection are less. Otherwise, if recipient receive a graft with MHC molecules which are having lot of mismatching, then for the survival of the graft, you have to do significant immunosuppression for a long time, sometimes even lifetime, for the person who is receiving the mismatched graft. And I told you that this mismatching, right, between the recipient tissue and the grafted tissue is basically based on the concept of MHC class 1 and class 2 molecules. So today onward, when you think about class 1 and class 2 molecule, you have to remember three things. Class 1 and class 2 molecules are associated with effective antigen presentation. They are associated with predisposition to certain autoimmune diseases, and they play a very big role in transplant acceptance or transplant rejection. Another thing, class 2, these MHC molecules also determine how effective immune response you will develop against an antigen. Suppose a bacteria come to your body, and against that bacterial antigen you make class 2 molecule, but another person gets the same bacteria, but he does not make any class 2 molecule which matches with that bacterial antigen. So person who is making class 1 or class 2 molecules against certain antigen will develop better response against that antigens. Person who is not having any class 1 class 2 molecule against a specific antigen, that person will not develop proper immune response for that specific antigen. Am I clear? That is why that against the same microbe, different people develop slightly different degree of immune response. Is that right? And there is a link with allergy also. You know, some people, let's suppose I develop, unfortunately, such type of class 2 molecules which love to stick with the pollen grain antigen and present the pollen grain antigen. You know what's going to happen? I'm going to develop IgE antibodies against the pollen grain, and I have more susceptibility to develop type 1 hypersensitivity reaction or allergic asthma. But if there is my friend and my friend does not make such class 2 molecule which can process and bind the pollen grain antigens, do you think he has the risk of allergic asthma? Does not. Because I received the pollen grain, my friend also received the pollen grain, I did have some stupid class 2 molecule which stick and process and present the class 2 molecule, and he does not have those stupid class 2. So his body does not activate the immune system against the pollen grain. My stupid MHC class 2 molecule bound with the pollen grain antigen, presented them to T helper cell, and then T helper cell become active and make maybe IgE against the T helper cell, activate the B cell to convert into plasma cell, which make IgE antibodies against the pollen, and those antibodies get loaded on the mast cell, and I'm the unfortunate guy having mast cells loaded with anti-pollen IgE. And who was the culprit? That class 2 molecule which very effectively and proudly presented the pollen antigens to my immune system. And later on I will have troubles. That's all for today. Class dismiss.